And then I'm going to introduce our next speaker. We're going to be hearing from Camilla Leask with Alaska Department of Fish and Game. They've been using Sample Manager for over a year, and today Camilla is going to be going to share how they've implemented it in the world of wildlife health and veterinary sciences. So they're managing samples in this field comes with unique challenges, and I'm excited to hear more about their experience. So please welcome Camilla.
Hello, let me get my screen sharing. All right, so I'm Dr. Camilla Leask, and what I'm going to be talking about is how we've been implementing Sample Manager and some of our adventures in data and sample management that we have had here in Alaska. I work with the Wildlife Health and Veterinary Services at the Alaska Department of Fish and Game. Also, here at the conference today is one of my colleagues, Cassie Duncan, who has been just fabulous at both helping develop how we're using Sample Manager, but also making sure that we stay on top of getting all of our incoming samples into the program so that we can make use of all of its functionality. So who are we? We are part of the Alaska Department of Fish and Game. And what our department does is we work on zoonotic pathogens as well as wildlife disease monitoring. So we look at things that may be enzootic or epizootic. So looking at rabies monitoring throughout the state, other diseases that may be important. When I have there the disease du jour, that's because there's always new things coming up. Currently, we're very focused on looking at high path AI. We're also involved in doing sampling and looking at how SARS may be affecting wildlife or in our populations. In the past, we've had investigations looking at Mycoplasma pneumoniae. So these are always changing, and that's always making us have to change direction and potentially even have to go back into our archives to find samples that would be appropriate to test to be able to track how these pathogens are moving through our populations. Our group manages an archive of frozen samples, also some that are not frozen, as well as a lot of the data that has been collected over the years for Fish and Game. We offer diagnostic and forensic services. So when there are mortalities, we will do necropsies and do samples and try and come up with why that animal was found dead. We provide some herd assessment tools. So we're looking at various things that we can measure to help better understand the health of the animals here in Alaska. We also are very involved in staff education and public outreach to let people know what's going on with our different populations. And we coordinate with various agencies, our state, federal, local, and academic partners, as well as international partners. So we have a number of needs in our group. We need to be able to store and track samples that are collected both from those live captures that the biologists are going out and doing, as well as necropsies that we are doing in-house. We currently have samples that have been archived since about 1968 to the present. We have things that are stored in ultralow and cryovials. We also have walk-in freezers and a lot of samples that are stored in various containers that are currently in totes. And then we also have samples like hair samples and tooth samples that may be in a cabinet or stored someplace else. And so we need to make sure we know where we find all of those samples when we need them. Currently, we have more than about 400,000 samples. And we also are a little Excel crazy. Over the years, we've used a combination of multiple access databases. Basically, we would create a new database every time we either had a new disease that we were investigating, or we have a different method or place that we're trying to store samples. because all of those different sample types or programs are going to have different requirements for what we need to track. We also have a large number of Excel files, and of course, the issue we have with those that over the year they can get corrupted. They can have errors that get introduced into them, and then we have to go back to old backup copies and recreate what we have done over a time period once we discover those areas, and it becomes very cumbersome and very time consuming. So our objective has been to have one database that can track all that information for easy access and retrieval. And ideally, we would like to have the ability to have those results that are also connected to the samples, in addition to being able to track those samples. And so some of the complications that we have come up with and issues that we have had when we have tried other programs is, and this is probably one of our biggest, is that the individual identification that we have for samples can be inconsistent, and a single animal may have many different individual identifications that it's given over the year. Because we have multiple sampling events where we might be capturing an animal many times over the years, um, sometimes each time it goes out and catches those sample, it may get a whole new ID. And sometimes during those captures, it might have the field ID that it's given for the capture. It might have a caller ID that's being tracked for that particular animal. It might be being used in another research project as well, so that project might have a number that's associated with that animal or that sample. So we end up with this crazy combination of IDs that we have to be able to track and manage and know which animal it came from, so that we don't have animals going into our database multiple times, even though it's all the same animal. We need to be able to store that metadata for each of those sampling events, because that can change. So for instance, at one point when we capture an animal, it might be in one area of Alaska, but because this is a migratory animal, the next time we see it, it may be in a completely different place. So sometimes even that simple thing of location of where that animal lives can be very broad, and we need to be able to know where it came from at any one time to better understand what we're seeing in those samples. Another complication we have is that we don't have all the information that we need for all samples. The data that we get in sometimes can be very sparse, especially for some of the older archive samples. So when a database requires certain fields to be filled, we can sometimes have difficulty getting those fields. So we need to have that flexibility to leave blanks where we have blanks, and then also to be able to go back later and fill that in as we learn more about that particular animal. Another thing that we have is that we don't do a lot of in-house testing. We have a few things that we look at in-house, but most of our testing is done in other laboratories. So we may be sending samples all over the United States and sometimes even internationally to get the tests done that we need. We also collaborate with a lot of people. So we may be taking subsamples that we send to others who are doing certain research projects, and we need to be able to track all of that. And we need to understand what we've tested on the different tissues already so that we don't have redundancy and accidentally send them out a second time. And also to know we periodically send the same sample out to different labs for similar tests in order to compare the labs and to have a better understanding of what we're seeing when we see discrepancies in some of our results. It can be very important to track related samples or animals. So for instance, if we have a calf, we may want to know what the maternal results were as well as the calf results. And so having the ability to track that relatedness within our database is something that's really important to us. And we also have a number of people over the years that have been putting in the data and updating things, and everybody has little quirks, and sometimes our labels may be, certain things may be abbreviated one way, and another time it may be abbreviated a different way. And so that ability to understand that all of these things are actually the same sample and having that consistency is really important, but also being able to maintain the ability to know what those vials are actually labeled as, so that if we're doing a search and in our database it says it should be one thing and the label says something slightly different, so that people who may not have a great depth of knowledge of, say, the biology, that those things may be the same, that they have a way of double checking that the label matches what the actual sample type is. And we also do a lot of searches that can have multiple levels and complications. So we might want to know how many samples do we have from a particular animal of a particular age in one region that has never been tested for a certain pathogen before, or what do we have, what are all the samples that we have tested for a certain pathogen, or where have we gotten certain results. So our searches can get fairly complicated, and so being able to get that data back out of our database is really important to us. We have tried over the years many different methods. I mentioned the Excel files and the Access databases. We did try another commercial database program that was for managing samples, and it was primarily designed for human medicine samples. And while it was good, it was very difficult to get our sample types and some of our complications to really work with that program. And on top of that, they were located in another country, and we had time zone issues, and just that kind of communication and trying to work through some of the problems just didn't happen. And then finally COVID hit, and their attention got put on trying to support all the places that were trying to deal with that particular outbreak, and support what was needed at that time. And we kind of gotten forgotten. And so we started looking elsewhere, and we ended up with LabKey and Sample Manager. And really, this list isn't very long, but it's a really powerful list for why Sample Manager works for us. And the big one is how flexible it is. It provides a set of tools, but how we define them and how we implement them is really customizable, and we can use it for all of our special conditions and weird things that we encounter. And as we find things that it doesn't do, when we talk to Lance and the people there, oftentimes we find out that that's something that they're already working on, or that there is an interest in adding those capabilities and kind of working with us and growing with us as we see what we need in a database. It does provide that all-in-one experience that we were kind of looking for, of being able to combine both our results and our samples and be able to track them all in one place. I will say that we are fairly early on in our adventure of getting all of that implemented. So far, we are mostly focusing on where the samples are and making sure that we can track that. And we are slowly implementing that storage of data, and are really excited about being able to have that and have access to that. So as we get questions and are trying to develop reference ranges for various assays, all that data is available to us. And I can't say enough about the last point, which is the excellent support that LabKey and Sample Manager provides. They answer questions extremely rapidly. They work with us in training us on how we might be able to implement some of the things we want, or they make a list of what it is that we have asked for and keep us updated as they implement things that may work for those solutions. So what are some of these features that are working for us, and how are we using them? I'm going to go over a couple of them right now. And the basic thing comes down to just our sample organization. What we've been able to do is create two different source types. And in this case, we have our animal, which are the things that aren't going to change as far as we know for that animal. So the species, and that we have in the sex for that animal. We can also have in that source all the different IDs that has ever been given. So as we get a new one, we have a number of fields that we can track and search that list all of those different IDs that it may have had over its lifetime. That way, when we get a sample in, we can do a search and see, is that animal already there? And be able to if it is already there at a new sampling event. And that gets down to our second source, which is that sampling event. And in that one, we can track what was it sampled for, how was it captured, how did those samples get to us. It can be where it was located and the age it is at a time. Because we do not necessarily have birth years for all of these animals, tracking that age that the animal was at the time of sampling is really important. And obviously that's going to change over time as well. And so that's why it's important for us to have those two different source fields and that we can't just all combine it into one source. And then lower down, we have our samples, and we currently have 6 different sample types that we can track. And then we can also look at aliquots or things that were split from those initial samples, which can be really important, especially when we're looking at samples, say a swab that might be tested for something like Mycoplasma ovonemoniae, where it can be really hard to get it out. We need to know if that was a serial sample, if that swab was done 2 different times, or if that swab was one swab that was split into a couple samples to send out to labs, because that can give us very different results when we're doing that analysis at the end. We have a couple special circumstances that can come up that we've come up with solutions to. So one of them is that one I mentioned for tracking cow and then fetus. So in this case, what we do is we make the fetus a sample type within the maternal animal. And that way, as results come back, we can track those results in tandem so we can see what came from the maternal side and which came from the fetus. We have the ability of going in and actually renaming those samples so that we can designate them as being fetal samples and not just from the mom itself. Another special circumstance that we have is if we go out and, let's say, get an environmental sample. In this case, we may have samples from 10 different animals that we are sampling. So that's 10 different animals with sampling events, but we might have an environmental sample that's going to affect all of those animals. And we can mark that in the database for how they're related so that as we get results back, we can see which animals are affected by that particular environmental sample. Which kind of gets into our sample organization for the different sample types that we have. And it's really powerful to be able to have these different sample types because each one is going to have different requirements that we need to be able to track. So what we need to record for a blood sample is going to be very different from what we might be looking at when we're dealing with, say, our organ tissues, where we're trying to describe exactly where we got it from. Is it a lesion? And we're going to need to track it in different ways. And so being able to have those different sample types has really helped us and cut down on the number of Excel files we need, because that's where we would end up having to do it in different files otherwise. So other things that we really use a lot are the pick lists. So once we have the tissues in storage and we're getting ready to send them to a different lab and we want to know what we're testing it for, we will actually add it to a pick list. So we can choose those samples. We can mark which lab it's going to. We have a set pick list for each of our common labs. And it'll be marked in testing for that particular sample type. That way we know which samples we're still waiting for results on. Our plan is to, once we get those results back, we can enter them into one of the assays. So we have the results in the computer as well and mark the tissue as consumed so that we can really track where it is. Once it's in that pick list, if it gets returned to us, because some of our samples get tested and then get returned, we can then immediately put it back into our storage and know exactly where it has been and really stay on top of that. We also utilize the jobs function for the workflow. Right now, we're primarily using it as a mechanism for people who are going in and they notice issues, but they may not have the permissions to actually edit or change those things. So they can flag the issues and let those of us who have that editing capability go in and fix those issues. We are in the process of creating templates that kind of make this easier. So for all of our common circumstances. We are also starting to set up creating templates for our sample shipping and updating results process so that we know, do we have those samples pulled? Have they been shipped? Where are they as far as the results go? Have we paid for those samples? And has all the results been updated? So we're really looking forward to being able to use that jobs function to kind of streamline and not lose track of what needs to be done when we have a whole lot of samples that get sent out all at one time. We are fairly early in our process. So far, we have entered what kind of our decision in transferring from our Access and Excel method of tracking to using Sample Manager is anything that came in starting the beginning of last year has been entered into LabKey, and we're slowly working through our databases of our older archive samples, making sure that there's no errors in what animals they are, making sure that we're not duplicating what we're putting into the database, but we're kind of slowly adding all of those archive samples. We're also working on getting those assays in. We're trying to decide how we need to set them up because we may test for organisms or things at many different labs and in different ways. We're trying to figure out the best way to organize those assays to get the results out that we need so that it has the most functionality for us. And then once we get all of those set up, we'll be starting to work the very slow process of importing all of our archived results. And one of the great things about Sample Manager and LabKey in general is how, I'm not going to entirely say easy, but it's a lot easier than our other methods. But how many different tools there are for kind of correcting and updating and importing. And so being able to import from a file in bulk has been fabulous for trying to get all of our data in there. When we do have an error, we can, so let's say an animal turns out to be had a different ID, turns out to be the same animal that we learn later on, we can very easily reattach those samples to the correct animal ID. We can change which sample events it's associated with for each of those samples without having to go in individually. We can do that in bulk. So there's a lot of tools that we have of being able to do some of those corrections that have been fairly laborious on our other methods that we have been using over the years. And that's pretty much what I have. Thank you for listening. And if anybody has any questions, I'm happy to take those.
Thanks so much, Camilla. I love to see all the pictures. That was my favorite part of your presentation, but that was really interesting, and I really do love the way that you guys have been able to sort of model the data. It is an unusual use case for us, so it's so cool to see that and to see that being successful for you. Please put in some questions in the Q&A or the chat if you've got some for Camilla. I'm curious about what are like the logistics of sample collection out in the field, and like how are you managing that, and then how does it get into the system, I guess. I'm just sort of curious how that works.
So biologists are doing their projects and they're collecting what they need. They will confer with us for if they have certain testing things they want. So we let them know what samples we need. But yes, trying to get those samples can be difficult. So depending on what species you're dealing with for captures, they may be darting them, they may be using traps. There's different things that they're going to be doing to get those animals and collect the samples. But this is one of the issues. If something, say, dies in a very remote place, just trying to get personnel out there to collect the samples and bring them back can be quite a challenge. And so sometimes the condition of our sample can be very interesting. And so being able to have a description and ways that we can track how poor or how good a sample is, so that we know what we can get out of it at the other end, or when an animal died, or when it was collected versus when we got it into our system, into our laboratory. Once we get it in our lab, we inventory it, and right now we can put it into an Excel file and upload it into LabKey to get those large groups. When we do a necropsy, we often do that by hand, where we are just entering the animal individually, because there's so many complicated levels to the tissues that we're collecting when we do those necropsies.
Got it. Yeah, that's really fascinating. Being in a much more lab setting where you're just, opening a specimen shipment box or something. It
Right.
feels a little bit like a different world of challenges that you
There's
guys.
a lot of planning that goes into what we do. I mean, we will send out totes that have all the pipettes and containers that everybody needs. We will often send personnel out to the lab to help process those samples in the lab or in the field setting. If we have samples that have critical needs, like need to be frozen immediately, we'll have liquid nitrogen out in the field to be able to do quick freeze on some of those. But yeah, that sample management in the field becomes a whole nother world.
Yeah, that's amazing. And then maybe just kind of curiosity around usage of Sample Manager. You showed that really cool lineage diagram of having kind of the sampling event on the registration. Can you talk about any positives or negatives, I guess, that you've seen with having to sort of register that intermediate source that, it is not sort of that associated with that physical sample that somebody's going in to register at the time?
Right. We get some challenges with, say, even just trying to figure out what species things are, because sometimes we're only going back one level, or it's telling us one thing, not another. But one of the things that we are gradually learning is how to do some of the filtering systems to pick those different levels. And so we're kind of in the early stage of learning that. But once we figure out that, if we can customize what we get out of our searches, we can start adding in some of those other levels, and see that. So we can very easily, if we care about, say, a certain herd, we can do a search for that herd, and we can go back and forth through the different levels to add that information in for the grid that we're getting for our results. So it works really well. But yeah, it did take a little bit for us to learn that, okay, we need to do our searches in a certain way to add that extra level of searchability.
Yeah, absolutely. Well, thank you so much again, Camilla. This was really great, and so fun to learn about on my side.